Now, today I would like to talk about our nanobio architect based on our DNA conjugate chemistry. Our DNA conjugate chemistry is shown in this slide. It was first developed for graft copolymer systems from polyanisopropyl acrylamide with DNA. In this case, this is very similar to the work by Dr. Osada, who talked just before, but in our case, core is made of hydrophobic polymer, synthetic polymers, and the hair or corona is made of DNA, natural polymer DNA. We prepared graft copolymer from polynipam main chain and about 10 branches of DNA, short DNA. If you raise the temperature over 37 degrees centigrade, polyanisopropyl acrylamide is dehydrated to give hydrophobic core, but DNA itself is highly hydrophilic and water soluble and negatively charged. So this type of nanoparticles are formed in aqueous system to give clear solution because the size of particle is about 50 nanometer in its diameter. But if you add the complementary DNA to the surface anchored single-stranded DNA, then to give double helical structure on nanoparticle surface, then the system becomes unstable to give turbid solution under the conditions of higher salt concentration, like 500 millimole of sodium chloride, like seawater. But if you add terminally mismatched DNA, such kind of aggregation did not take place. You can see clear solution, even in high salt concentration. I would say that one nanoparticle is made from about 20 polynipam graft DNA copolymers, and each graft copolymer has about 10 chains, 10 branches of DNA chains. This type of salt-induced non-cross-linking aggregation was observed not only for polymeric materials, but also for gold nanoparticles like this, having diameter of 50 nanometer. Gold nanoparticle modified with single-stranded DNA gives colloidal suspension like this, and the reddish color due to the monodispersed colloidal system was seen even at very, very high source concentration of 2.5 molar of sodium chloride. But if you add fully complementary sequence to this, you can see the color change at around 0.5 molar of sodium chloride. In contrast to this, terminally mismatched DNA did not give such kind of color change at all. So terminus-dependent behavior was also observed here. If you take TEM, transmission electron microscopic view of this colloidal system, you can see the aggregated nanoparticles over 0.5 molar of sodium chloride. So I said that gold nanoparticle, in this case, diameter is 40 nanometer, but anyway, fully matched double helical structure gave nanoparticle system unstable. But if the terminus is mismatched like T and T, then the colloidal system is very stable to give monodispersed one, and you can observe reddish color like this. So how about the second position mismatch? Then again, you can see reddish color. And how about the third position mismatch? This also gave reddish color. But fourth position mismatch, we cannot see reddish color, but you can see aggregated nanoparticle like this. So this colloidal stability change may be explained by the flexibility of double-stranded DNA surface. I mean, flexible terminal is breathing, and this would help dispersiveness. But if the terminus is rigid, then aggregation should take place. So the idea is coming, if you change the stability of stiffness of terminus, or you can, by using some target, or by using target-induced base pairing to stiffen double-stranded DNA terminus, then aggregation should take place. Due to the Professor Akira Ono's study, thymine-thymine mismatch can be base paired in the presence of mercury ion. This type of metal ion mediated base pairing with non-Watson-Crick manner can be beautifully utilized for mercury ion sensors with using this configuration of molecular beacon structure by Professor Ono's group of Kanagawa University. So this stable dispersion may be changed by the presence of mercury ion. So we added these three systems with mercury ions. Then the result is shown here. If TT mismatch is located at the end of the double helical structure, you cannot see any change. But when TT mismatch was located in penultimate position or second position, then you can see color change at 0.1 micromolar of mercury ion. And in the case of penultimate position or third mismatch, then you can see the color change at 0.5 micromolar of mercury ion. This type of responsiveness to mercury ion is not seen for other divalent cations. No color change was observed for copper, cobalt, or zinc or tin, like this way. So you see that if you change the stiffness of terminus by using some target ion, then you can control the stability of colloidal systems. Now, how about the physical stimulus, like light? Here I would like to show you photo-induced aggregation of double-stranded DNA gold nanoparticle systems. If you can change the stiffness of double-stranded DNA terminus by using some physical stimulus, then you can make you can change the colloidal stability remotely. And for this purpose, we took advantage of good reagent presented by Professor Asanuma of Nagoya University. Professor Asanuma developed the azobenzene modified nucleotide model, and he found that transform of azobenzene structure can stabilize the double helical structure of DNA. But if it changes to cis form, then cis form disturbs that double helical structure, and duplex is dissociated into two single strands. For this purpose, Professor Asanuma introduced several moiety of azobenzene moieties into middle region of DNA structure. In our case, we introduced one azobenzene Asanuma compounds into just before the terminus, very close to the terminus. And in the transform, the photostationary state transform, we can observe aggregated nanoparticles, and you can see purple color like this. But if you add UV light to this cuvette, then color change, you can see color change to give reddish color. This means nanoparticles are dispersed with this type, this photo irradiation. And this change was fully reversible. With the irradiation of UV, then you can see a reddish color. And if you irradiate visible light for 10 minutes, then you can obtain purple color due to aggregated gold nanoparticles. Then again, UV light to give reddish color, and then purple color with visible light. As I explained, gold nanoparticles can be controlled to give aggregated state with complementary DNA and keep dispersed state with the addition of terminally mismatched one. But aggregated structure is like this. How can I say this is kind of amorphous, no interesting structure? This is not a so-called nano architecture. So we are interested in a bit more sophisticated structure, and we prepared a relatively simple one, 3 nanoparticles on 1 template DNA. And in this case, we can also see shrinkage for complementary DNA, and no change was observed for terminally mismatched DNA. This is one of the results. In the case of full match DNA, you can see the interparticle distance about 10 nanometer. But in the case of addition of terminally mismatched DNA, then you can see 14 nanometer of interparticle distance. This is also probably due to the aggregation and the dispersion difference. This type of 3 beads, 3 nanoparticles on 1 chain can be prepared like this way. Procedure is a bit complicated, so I would like to show you just a scheme. As for the template, we prepared 200 nucleotide DNA, single-stranded DNA. And here are 3 positions which can be bound to gold nanoparticle. This yellow part, 16 nucleotide part, which is complementary to surface-anchored DNA on the gold nanoparticle. Such template binding DNA is only 1 on 1 nanoparticle, and the other 5 DNAs have this type of sequence, which is used for responsive DNA. I mean, these 5 DNAs are full matched or mismatched. And the result was like this, the former one. In the case of full match, you can see shrinked one, and terminally mismatched one, you can see elongated beads on a string nanostructure. Now, I showed that the stimulus responsive structural change of linear trimer case. And we prepared quatermar, 4 nanobeads on 1 string. But it is not easy to make this type of structure bigger and bigger. So we changed our mind for obtaining long linear nanostructure with using a very long template DNA. For this purpose, we used rolling circle amplification procedure to obtain very long DNA having nanoparticle binding domain like this way. And the result is shown here. For when you add full match DNA, then you can see this type of island structure. And the interparticle distance was about 5 nanometers. And in the case of terminally mismatched DNA, if you add terminally mismatched DNA, you can see this type of elongated structure, and the interparticle distance was longer, like 9 nanometers. So terminal structure dependent behavior is also observed for this type of long linear nanostructure. Now I'd like to move to the other nano architecture system, stimulus-responsive structural change of gold nanorod assembly. Gold nanorod has been studied in detail by many scientists, and Professor Murphy reported first the modification, selective modification of gold nanorod surface, and Professor Kotov reported recently the modification of nanorods by using with DNA, and his finding that is that DNA thiol can preferentially bind to the both ends of gold nanorods, and if you modify the side region, you need a bit more higher concentration of thiol modified DNA. So with using his idea, we modified the surface of gold nanorod with one DNA with relatively low concentration and the second DNA with relatively high concentration. So DNA number 1 is immobilized at both ends, and DNA number 2 is immobilized on side surface. Why we would like to make this type of differential selective modification? What we would like to do is this type of structural change using mismatch and full match structural change. If you want to prepare side-by-side assembly, full match DNA should be located inside, and the top and bottom should be mismatched. But in the case of end-to-end, for the case of end-to-end assembly, full match structure is needed at both ends, and the mismatch should be immobilized on side. And this was really true in the case of full match in the side region. You can see this type of side-by-side assembly. And in the case of in the case of full match at both ends, then you can obtain this type of end-to-end assembly. In this case, side region is modified with mismatched DNA, TT mismatched one, so you cannot see any side-by-side assembly on these pictures. So how you can change the side-by-side to end-to-end assembly by using some chemical stimulus? We can use this type of mercury ion mediated thymine-thymine mismatch to thymine-mercury-thymine non-Watson-Crick base pairing. This is yes gate. So you end-to-end assembly is prohibited for this case, but you can, if you use this type of reaction, you can make end region to be adhesive to give end-to-end assembly. But it is not enough. You have to change the side-by-side assembly non-effective. This is on switch, for example, but you also need off switch. You have to change the sticky interaction to non-sticky one. For this purpose, we used NOT gate. For this, with using this structure, aggregated nanoparticle can be changed to disposed one. What is the essence of this NOT gate? We used inside Barge structure like this. In this case, AT full match is formed to give turbid one or aggregated one. But with the addition of mercury ion, T-mercury-T base pair is formed to give single base protruding overhang of A. This A is flexible, and this flexibly moving A can stabilize the colloidal system. Actually, this worked like this way. So with using this NOT gate, we carried out side-by-side assembly to end-to-end assembly. And this was the result. Side is fully matched TA, but this is a kind of NOT gate. And the terminus, both ends are mismatch, TT mismatch. Then the result with the addition of mercury ion, both ends should be stabilized to give sticky ends, but the side should give protruding A to give non-sticky surface. However, the result, first side-by-side assembly was observed, but with the addition of mercury ion, then you can see end-to-end assembly like this way. Now, I have prepared some other slides, but it's about time. Oh, I have 4 minutes, so you may wonder how this system can be used for genotyping. I don't have to explain in detail as to this SNP genotyping, but it is important to know the one base difference for realization of personalized medicine. For this purpose, this strictly recognizing behavior of terminal structure to change the structure of colloidal systems, this would be good for application of SNP genotyping. But it was not true. In the case of X equal A and Y equal G and T, AT should change the color, but AG changes also the color. In the case of X equals C, only G changes the color. This is okay, but this results in some mistyping. But this is actually possible because AG can also make a kind of non-Watson-Crick base pairing. So this should result in mistyping. So we changed our mind. We used one base protruding should result in color change. One base protruding should result in the stabilization of nanoparticle system. To utilize this, we used single base extension reaction. And for this purpose, 16 primer is used just before the SNP position. And now, if the SNP position is A, then only T should react. Dideoxythymine triphosphate should react to give one base extended one, and the other three did not make reactions. So, and if you add gold nanoparticle having the surface anchored DNA which is complementary to the primer, then 3 bottles give purple color, but only DDTTP containing tube makes nanoparticle color change to reddish because of one base extension of T. And this was good for wild type sequence and mutant type 1, and heterozygous model also showed good result. And actually we used 3 persons hair to check SNPs of each person, and this is the result. Wild type, and mutant, and this is the result of DNA sequencing. This endorsed the results of our test tube systems. And this shows similar results can be also observed for nanorod and nanoplates in addition to nanosphere. These structural difference gave the difference in their colors, gray, blue, and red. This type of multicolor different systems may be used for more sophisticated diagnostic systems. We are now working on this. Now, before closing my talk, I'd like to thank my colleagues at the Riken Institute, Dr. Takarada, Dr. Akiyama, Mr. Shiraishi, and Dr. Wan and Dr. Kanayama have worked very hard to make a very good contribution to this talk, my talk today. And finally, I'd like to thank your kind attention. Thank you very much.